Hello, my name is Yao Chen. I'm an assistant professor in neuroscience at Washington University in St. Louis. My lab studies two major directions. One is neuromodulator actions, the other is sleep functions, and I'll tell you about both. We are fascinated by neuromodulators. These are chemicals made in the brain, for example, dopamine, serotonin, and acetylcholine, and they can have transformative effect on our behavior, regulating, for example, emotions, attention, learning, and memory. And neuromodulators are not only critical for healthy conditions. Neuromodulator receptors are targets of almost all current drugs used to treat psychiatric disorders. However, we don't understand how neuromodulators can transform behavior under healthy conditions, and we also have very little idea about how drugs targeting neuromodulator receptors can help alleviate psychiatric disorders. And we know even less about why these drugs can have beneficial effects for some patients and very little effect for others. So we really want to understand how neuromodulators function in normal and disease conditions. There's a major paradox surrounding neuromodulator actions. There are many neuromodulators. In fact, there are hundreds of neuromodulator receptors. They transmit signals from the outside to the inside of the cell. However, inside the cell, these hundreds of receptors converge onto very few signaling pathways. Yet, these receptors can impart different functions to the cells and to the organism. So how can hundreds of receptors converge onto very few signaling pathways, yet have different functions? And our research is motivated by the idea that it's not just the identity of the signals inside the cell that matters. What really conveys the information is where these signals inside the cell are active, what kind of temporal patterns do they show, and how do different neuromodulators interact with each other so that the same neuromodulator can have different or even opposite effects depending on the cellular context. And we have developed special technologies and built special microscopes to capture these signals inside the cell in real time. And we are using these technologies we have developed to understand how the spatial, temporal, and context-dependent features really convey information and how that information is used to alter cells, circuits, and eventually behavior and emotions. For the second direction in my lab, we are fascinated by sleep because all of us spend about a third of our life sleeping. Yet we understand very little about how sleep has many physiological and behavior functions. We all know sleep is important for good immune systems and for healthy emotions, for learning and memory and alertness. Yet how does this state of sleep produce so many important functions? Because our lab has special technology and has insights from our study of neuromodulator research, we figured that if we can watch signals outside and inside the cell in real time and figure out which signals are different between sleep and wakefulness, that will immediately give us hypotheses on what could be the cellular functions of sleep. And we hope that's a string that we can keep pulling to go from the cellular functions to understand how sleep imparts circuit functions and organismal functions. So these are the two aspects of our lab, and we try to create a lab that's kind and creative, rigorous, and resilient. And we would love for you to join us. Feel free to contact me if you have any questions about our research about our lab. Thank you.
Hello, I'm Pingchuan Ma. I'm a PhD student from Dr. Yao Chen's lab. I'm from China. Currently in the lab, I'm working on a project that extends the usage of neuromodulator biosensors into our specialized imaging method called fluorescence lifetime imaging. Neuromodulators are critical to our brain functions and behaviors. The usage of biosensors enables us to monitor the dynamics of neuromodulators in the nervous system. Our measurement of fluorescence lifetime is independent of the expression level of biosensors, many other imaging conditions, and hopefully the heterogeneity across different animals. So our imaging method will be perfect for long-term recording of the neuromodulators. Our lab is a neurobiology-focused lab, but working on the interface of bioengineering, biochemistry, optical physics, and even computational work. So welcome to explore more about our lab. Thanks.
Hi there, my name is Kush Qin, and I'm a second-year graduate student in Dr. Yao Chen's lab. I come from China. I'm currently working on exploring the subcellular localizations of neuromodulator receptors in hippocampal neurons using CRISPR-Cas9-based genome editing system. The subcellular localizations of neuromodulator receptors have fundamental impact on the cell signaling pathways. So understanding the localizations of different types of neuromodulator receptors could help us review the mechanisms underlying the complex cell signals. I like the atmosphere in Chen Lab that everyone is so passionate to explore the mystery of neuromodulators, of how they're modulating our nervous system and further modulating behaviors. Welcome to WashU and welcome to St. Louis. I hope to see you soon.
Hi, my name is Peter Chen, and I'm a master's student working in the Chen Lab from Houston, Texas. I'm currently working on a project where I'm creating a photoactivatable enzyme inhibitor peptide. When paired with optogenetic stimulation, these peptides would allow us to control the availability of specific enzymes with high temporal precision. The ability to control enzyme activity then allows us to study their functions. My favorite part about the Chen Lab is the open and intellectually stimulating environment it provides. Everybody is always happy to collaborate or talk to you about their work, and that allows you to receive constant feedback about your own projects and also learn about a wide array of topics and techniques.
Hello, and welcome to WashU and to Chen Lab. I'm Anna, originally from Sweden and with a background in dentistry, and my role in Chen Lab is being the lab manager. And I'm trying to spend my time helping out on the different activities that are going on in the lab and trying to get the lab to run as smoothly as possible from day to day. So that's about it, and I hope to see you around in the future. Bye.